Let us start today's lecture. So, today's topic of discussion is about the pancreatic hormone. So, mainly we will try to focus about the synthesis part first, then mechanism of secretion, and after that I will discuss about the signal transductions that is involved with the actions of insulin hormones mainly. So, at the very beginnings, let us start with some introductory informations about the pancreatic glands and pancreatic hormones. See here, if you see the anatomical features of the pancreas, it is a specific locations, and this pancreatic glands or pancreas are involved with the secretions of a certain type of hormones, and the hormones release or synthesis or secretions basically occurs for specific cell types. Now, question is, what are the most important cell types that involves with the secretions of important pancreatic hormones? So here, at the very first glance, it is mentioned that it is very special glands because it has two different property, right? It has exocrine functions as well as endocrine functions, right? So we will see what are the most important type of exocrine secretions and what are the endocrine secretions, and how these both the exocrine and endocrine secretions are equally important for cellular functions. The most important parts of these endocrine glands is discrete islets of Langerhans. Why it is important? Because it contains different type of cells that mainly play an important role for the secretions of important metabolic hormones. So, what are the most important cell types? It is mainly composed of 5 different endocrine cell types, right? So, what are these? These are alpha, beta, delta, epsilon, and epsilon. So, all the different type of cells that are present in the islets of Langerhans secretes a different type of hormones. You can see what are the most important type of hormone secreted by these cell types. Like they secrete 5 different type of hormones. So, what are these? These are glucagon, insulin, somatostatin, ghrelin, and pancreatic polypeptide or pancreatic peptide respectively. It means the alpha cell secretes glucagon hormones, then beta cell secrete insulin hormones, then delta cell secretes somatostatins, epsilon secrete ghrelins, and epsilon secretes pancreatic polypeptides, right? So, all those hormones are equally important for a physiological point of view. Now, before proceed to the main mechanism of hormone actions, we first will try to discuss about the most important type of hormones, that is about the insulin hormones. You can see about the histological features of this, mainly the most important cell types, like what is the features of the alpha cells and what is the features of the beta cells and what is the features of delta cells. If you see this structural morphology of cell types, yes, there is a morphological difference of cell types. Not only morphological difference, at the same time, with the changes in the morphology, there are changes in the secretion patterns too, right? Like alpha secretes the glucagons, beta secretes insulin, and the delta secretes somatostatins. If you see the most important cell types that is present in the external portion of the pancreas are SNS cells and duct cells. Both the SNS cells and duct cells are most important because they secrete some enzymes, so-called digestive enzymes. Now, in today's discussions, I will try to explain about the insulin first. After that, I will proceed the glucagon part in the next verse. See here, the most important type of enzymes that is secreted by SNS cells is trypsins and amylase. Both the trypsin and amylase equally important in the digestion process. And at the same time, if you see some cell that is the F cells, so-called pancreatic peptide cells that secrete some peptide hormones. Now, if you say about the importance of insulin hormones, yes, we can say insulin is very much important for us because it regulates the blood sugars. Now, question is, what is the exact mechanism of insulin synthesis? Whether insulin synthesis, whenever it's synthesized as active forms or not. If not, how actually it processed. If you see the features of this insulin hormone synthesis, the insulin mRNA is translated as a single chain precursor, and that single chain precursor is nothing. This is the preproinsulin, right? What is the features of this preproinsulin? If you see the structures of this preproinsulin, it is composed of different segments. All the segments of preproinsulins are not functional. Some of the segments are functional, some are non-functional, right? And this is a polypeptide chain actually. And in this polypeptide chain, there is an N-terminal end and another is C-terminal end, right? So here, what are the most important segments? Most important segments are that initiates with the signal sequence, and after that there is a sequence of B segments. Then this is called the C-peptide or connecting polypeptides, and after that it contains the A sequence. And all these segments are mainly composed of different types of amino acid only, right? So here, this preproinsulin is not the active forms. So to become active forms, it must go to some catalytic events. So in these catalytic events, some important enzymes involved in the catalytic process. Here you can see the first event that takes place is the cleavage events. The cleavage causes the separation of this signal sequence from the preproinsulins. Another important event that takes place is the formation of the disulfide bonds. So the disulfide bonds basically occurs between the B segments and the A segments, right? So during the process of this preproinsulins, it removes the segment C or connecting polypeptide chains, right? And ultimately, it forms an active form of insulins. Now, as I mentioned, the initial points of time, the insulin that is formed is not the active form, which is an inactive form. To become an active form of insulin, it must pass through some biochemical events. So in this event, cleavage takes place, and there is a formation of some functional chemical bonds, mainly the disulfide bonds, and ultimately they produce the active form of insulin, and by removing the connecting polypeptide or C-peptides. Here you can see the events that occurs at a particular sites of cell organelles. You can see the most important type of cell organelles that involves the maturation of the insulin is the endoplasmic reticulums and the Golgi apparatus, right? And another important information that I want to mention here, the half-life of both the connecting polypeptides and the insulin hormones. If you see the half-life of both the C-peptides and insulin hormones, here you can see the half-life of insulin hormones is around 4 to 6 minutes, and the half-life of C polypeptide is act as a insulin markers. It acts as markers of insulin synthesis as well as the secretions. Now, after the synthesis of insulin hormones, what next? The next event is it must release, right? So we have a specific secretory mechanisms of insulin. So what is the mechanism of insulin secretions, or what are the influencing factors that helps in the insulin secretions? You can see, before proceeding to the secretions, you can see some important points that I have mentioned here. The half-life of insulin in naturally produced hormones is very short, and it is only about 4 to 6 minutes. And for metabolic point of view, the half-life is very important, right? It actually, the half-life of particular hormones actually has to respond with the metabolic changes or the changes in the changes of particular biomolecules in a circulating blood. You can see here, the short half-life is necessary for the body to quickly respond to the changes in the blood glucose levels, right? And yes, so to maintain the optimum half-life, or to maintain the optimum amount of insulin hormones in the circulating blood, basically depends on a particular organs, that is liver. So what is the role of liver here? The liver plays a significant role in insulin clearance. and it was found that around 50 to 60% is being extracted by the liver before it reaches systemic circulation. It means whatever the amounts of insulins are present in the circulating blood, it is not the 100% insulins. Before reaching to the circulating blood, these insulin hormones are degraded by the liver, some liver enzymes. So that is why liver is important for clearance purpose. And even if you see some research study, according to a research study, the liver is the primary site of insulin degradations. So it means some informations are given here. So what are the most important informations? The important informations are whatever the active form of insulin are synthesized. Not all the active form of insulins are found in the circulating blood. Before reaching to the circulating blood, it may undergo the degradation events, and these degradations is carried out by some liver enzymes. And here you can say this is some established by some research study that the liver is the primary site of insulin degradations. Now, if I ask what is the most important target site for insulin hormones and what is the most important functions that is carried out by insulin hormones. If you see this target site, yes, the most important sites are hepatic muscles as well as the adipocyte cells, right? These three are the most important target site for insulin hormones. But it is not limited to these cells only. Other target sites also this for insulin hormones can act, but these are the most important target sites. So what is the important functions that takes place in those target sites by insulin hormones? You can see in presence of a sufficient amount of insulin hormones from beta cells, it targets the liver cells, mainly hepatic cells. It reduces the biosynthesis of glucose, or it reduces the glycolytic events. And not only this, it also promotes the glycogen synthesis and accumulations of lipids, as well as it also increases the rate of inflammations in the liver tissues. If you see the peripheral muscle cells, what is the role of these insulin hormones? The most important rules in peripheral muscle cells, it involves into the glucose metabolisms, it involves in the glycogen biosynthesis, and other than this, it also takes part in the increase in the muscle mass. And due to the presence of insulin hormones, what we have is there is some dysfunctions of mitochondrial functions. So it means these insulin hormones, in one way, it involves the metabolic process like metabolism of glucose and metabolism of glycogens. At the same time, insulin also takes part in the dysfunctions even to the mitochondrial functions. Now, if you see the role of insulin hormones in the adipose tissue, here also it increases the glucose metabolisms, and the most important things in the adipose tissue that is facilitated by insulin hormones is lipogenesis, right? And it reduces the lipolysis. So these are the most important functions that is carried out by insulin hormones in the target cells or target tissues. As I mentioned, the actions or functions of the insulin hormones are not limited to this target tissue only or target cell only, it also acts into some other cells too. Now, see the most important part of the discussions. As we have already mentioned that the beta cells, the most important site of insulin hormone synthesis, and these insulin hormones acts when it matches into the active form of insulins, and it was found that only 50 to 60% insulins are circulating into the circulating blood. Before enters into the circulating blood, it is degraded by the liver enzymes. Now, whatever the amounts of insulin synthesized that must release to the released from these particular glands and acts to the target cells. So, what is the exact mechanisms of insulin hormone release, or what is the regulatory mechanisms of insulin hormone secretions? See, the most important things, the availability of the blood glucose. Plasma glucose level is the main regulator of insulin secretions. It means it is the influencing factors, most important influencing factors we can say. The change in the concentrations of plasma glucose that occurs in response to feeding or fasting is the main determinant of insulin secretions. It means these are the two conditions, feeding and fasting. So both the conditions alters the level of blood glucose, and alterations of blood glucose levels ultimately lead to the changes in the insulin secretions, right? Now, modest increase in plasma glucose levels provoke a marked increase in plasma insulin concentrations, right? So here again it is mentioned, if there is a certain changes in the plasma glucose levels, there is a marked increase in plasma insulin concentrations. Glucose is taken up by beta cells via glucose transporters. Yes, there are certain types of transporter molecules that are basically present in the cell membranes, and those transporter molecules allows the glucose molecules. Here, during the regulations of insulin secretions, the most important types of transporter involves the GLUT2 transporters, right? So, what is the role of GLUT2 transporter here? It allows the influx of glucose into the circulatory blood. Here, the glucose is taken up by beta cells first by the glucose transporters, and after that there are certain changes takes place in the cells. What are the changes that takes place in the cells? See, the subsequent metabolism of glucose increases cellular adenosine triphosphate concentrations and closes ATP dependent potassium channels in the beta cell membrane. So two things happen here. The first thing is the glucose that is taken up by the beta cells by certain transporters, that is GLUT2. After that, you see, the subsequent metabolism of glucose increases cellular adenosine triphosphate concentrations. So once there is changes in the concentrations of ATP along with the certain other changes like changes in the potassium channels that is present in the beta cells, causing membrane depolarizations and influx of calciums. It means, you can see from here, this is the transporters, that is the GLUT2, and this GLUT2 transporter allows the glucose molecules, and this GLUT2 transporter basically present in the beta cell membrane, right? Once the glucose enters into the cell cytoplasms, it undergoes through a metabolic events called glycolysis, right? When it enters into the cells, beta cells, it undergoes the glycolytic events and ultimately leads to the formations of ATP, because it not only completes the glycolytic events, it also undergoes to the Krebs cycles and cellular respirations. Ultimately, it produces the ATP. Now, where the level of ATP is more into the beta cell cytoplasms. So, as I have mentioned here, the subsequent metabolism of glucose increases cellular adenosine triphosphate concentrations. So once there is changes in the ATP, it alters the activity of the potassium channels. See here, this blocks the potassium channels, right? This higher level of ATP blocks the potassium channels, right? So it prevents the efflux of potassium ion. Now, due to this changes in the potassium ion channels, there is a changes in the potential of the beta cell membrane. So what is the changes? See, it closes the ATP dependent potassium channels in the beta cell membranes, causing membrane depolarizations, means the changes in the potential in the beta cell membranes. There is a potential change, because whenever it blocks the potassium efflux, at the same time it allows some other molecules like sodium ions and calcium ions. So due to the influx of this positively charged ions into the beta cell cytoplasms, there is changes in the membrane potentials called depolarizations, right? Now, this depolarization events or the calcium influx is the most important ones here. So, what are the other changes that takes place? See, increased calcium or intracellular calcium results in an increased insulin secretions, right? So, ultimately, in these particular conditions, there is a higher level of calcium ions. This higher level of calcium ions triggers the insulin secretion process, right? See here, This insulin are basically found in a vesicle structures, right? Some of the insulins are present as a reserve poles, some of the insulins are present ready to releasable poles. So here, both the case, the release or secretions of these insulin that are present to the vesicles or that are in the reserve poles, that is influenced by the availability of the calcium ions, right? Once there is a higher level of calcium ions into the beta cell cytoplasms, it stimulates these vesicles to release from the ready to releasable poles. Ultimately, there is a release of insulin hormones, right? Here, the release or secretions of these insulin hormones or the rate of secretions of insulin hormones are influenced by the availability of calcium ions. At the same time, it is also influenced by some other state. So, that can be explained by some phases. If you see the phase of insulin hormone release or insulin secretions, it can occur in two different phases. The first phase and second phase, called biphasic release of insulin or biphasic secretions of insulin hormones. And these biphasic secretions of insulin hormones are influenced by different factors. Like at the initial point of time, like for the first 4 to 6 minutes, there is a rapid secretion of insulin hormones, right? Because in this particular situation or in this particular conditions, there is a more amounts of calcium ions presence. At the same time, this also triggers by some other factors. These are the some other factors that amplifies the formations of ready to releasable poles of insulin that allows the rapid efflux of insulin hormones, right? After completion of first 6 minutes or 4 to 6 minutes, it passed to a sustained phase of insulin secretions, right? This is called the second phase of insulin secretions. It means the rate of insulin secretions are influenced by mainly two things: the availability of calcium ions. At the same time, it can be amplified by some other factors. These are some other factors that amplify the pathway of insulin secretions. This is what is the biphasic secretions of insulin hormones. So, you can say the initial phase, the secretions is very rapid and it continues up to 6 minutes. After that, there is a sustained secretion of or it maintains a steady level of insulin secretions. Now, see here, what is the most important type of transporters involved? That is the GLUT2 transporters. And these GLUT2 transporters allows the glucose molecules. So, here ultimately the secretion of insulin hormones, we could say the secretions of insulin hormones is mainly depends on the glucose uptake by GLUT2, right? That is explained here. This is the glucose uptake by GLUT2 transporters that undergoes the cellular aspirations that ultimately produce the ATP, and that ATP blocks the potassium ion channels. Instead, they promotes the sodium and calcium influx and ultimately leads to the changes in the potential of beta cell membranes, so called depolarizations of the beta cell membranes, and that allows the rapid efflux of the calcium means insulin hormone from vesicles to the outside of the cells, means into the circulating blood. Now, here you can see glucose is the main stimulator of insulin release from the beta cells, which occurs in a characteristic biphasic patterns, an acute first phase that lasts only a few minutes, followed by sustained second phase. This is the most important things. Now, at the same time, if you see the insulin secretions, it also can be explained by some other ways, like it can be explained by glucose insulin dose response curve, right? So, what is the meaning of glucose insulin dose response curve? See, the shape of the glucose insulin dose response curve is determined primarily by the activity of glucokinase, which governs the rate limiting state for glucose metabolisms in the beta cells. Means through the GLUT2 transporters, there is an influx of glucose, and this glucose undergoes the glycolytic events. So, the glycolytic events, in the glycolytic events, there is a special enzymes, right, called glucokinase that catalyze specific steps of glycolytic pathway called rate limiting steps. So, here these rate limiting steps actually important here, because these rate limiting steps decides about the amount of ATP formations into the beta cells, and at the same time, if the amount of ATP formations is less, obviously the potassium channel functions will be of different types, or patterns will be different, right? That ultimately will affect the secretions of insulin. So here the glucose insulin dose response curves played an important role, and that can be explained by the level of glucose into the circulating blood. You can see glucose level below 5 millimole per liters do not affect insulin release, right? But half maximal stimulation occurs at about 8 millimole per liters. So up to these amounts of glucose doesn't have any effects in the insulin secretions, but once it reaches the 8 millimole per liters, it stimulates the release of insulin secretions. So this can be seen from here. You can see this is the glucose insulin secretion curve, or so-called dose response curve. Initially, you see the secretion levels of insulin is very less. Here the plasma glucose levels is less than 8 millimole per liters, right? The secretions of insulin is also very less. But once it crosses this level, there is a increase of insulin secretions, right? So it means initially up to 8 millimole, 5 millimole per liters, there is no such changes in the insulin secretions, but once it reaches to 8 millimole per liters or more than that, there is a rapid increase of insulin secretions. So in this slides, two things are discussed. One is the first phase of so-called biphasic insulin secretions. One is the first phase, and another the second phase. The first phase sustains for 5 to 6 minutes or 4 to 6 minutes. After that, there is a sustained secretions of insulin hormones. So initial point of time, there is a rapid secretions. Now, after the secretions of insulin hormones, the insulin hormone must act into the target cell, right? Now, question is, what is the mechanism of insulin hormone action? Yes, if you see the insulin hormone actions, it is little bit complicated compared to the other hormones, right? See, insulin binds to specific receptors. The name of that receptor is receptor tyrosine kinase, and that receptor tyrosine kinase is present into the plasma membranes. So once this insulin binds to the receptor tyrosine kinase, there is an increase in the activity of phosphorylation events, and that phosphorylation leads to the regulations of different type of enzymes and different type of protein substrates. So here's the most important thing that I have mentioned, the type of receptors that played an important role in the insulin hormone actions, that is called receptor tyrosine kinase. So now question is, what is the structural features of this receptor tyrosine kinase? Whether this receptor tyrosine kinase is same structure is same to other receptors? No, it is little bit different, right? See here the structural basis of receptor tyrosine kinase first. So why receptor tyrosine kinase or insulin receptor tyrosine kinase is important? Because this insulin receptor tyrosine kinase is very, very specific for insulin hormone only, right? And it is some other property, like it has some autocatalytic property, right? This insulin receptor tyrosine kinase has some autocatalytic property. So what is the features of these receptors? Obviously, like other receptors, it is present into the cell membrane. It is composed of some subunit. So here's the most important subunits are alpha subunits and beta subunits. If you see the alpha subunits are basically present in the extracellular side of the cell membrane, right? This is the extracellular side of the cell membrane, right? Whereas the beta subunits are traverse, and some of the part of the beta subunits present in the extracellular surface, some parts traverse the cell membrane, and some parts are present in the intracellular surface of cell membranes, right? As I mentioned, it is some special property called autocatalytic property. How and why? It is autocatalytic property because it itself can phosphorylate. So one receptor phosphorylates just to opposite one, and vice versa. It means there is a cross phosphorylation events takes place. Where actually this phosphorylation takes place? It takes place in the tyrosine kinase domain of this insulin receptor tyrosine kinase. See, this is the tyrosine kinase domains. In this tyrosine kinase domains, there is a special amino acid sequence, that is the tyrosine amino acids. And this tyrosine amino acid is the site of phosphorylations. Yes, for these receptors or for this tyrosine domains, the phosphorylations is carried out by this tyrosine kinase domain, and for this tyrosine residue, phosphorylations is carried out by this tyrosine kinase domain. It means there is a cross phosphorylations of insulin tyrosine kinase receptors, and here this cross phosphorylations is carried out by tyrosine kinase itself. That is why you could say this insulin tyrosine kinase receptors is a autocatalytic receptors. Now, as I mentioned, this is very specific, and this autophosphorylations is basically depends on the proper binding of insulin hormones, right? Once insulin hormone binds, it undergoes two different signaling cascade, right? What is these two different signaling cascade? The two signaling cascade is, one is MAPK pathway and another one is PI3K pathway, right? See, this is the first pathway that is activated by the binding of insulin hormones to the insulin receptors. This is called MAPK pathway, and another one is PIK3 pathway. Here, let me just brief first. Here, insulin signal means once insulin binds to the receptors, there is autophosphorylations of these receptors, and autophosphorylation leads to the activations of these receptors. Once these receptors get activated, it undergoes through two different cascades. The first cascade is this cascade. So, in this cascade, what is the most important thing? It activates the insulin receptor substrate. So, if you see the characteristics of insulin receptor substrate, insulin receptor substrate has some special domain called SS2 domain or sulfide domain. Due to the presence of this SS domain in this insulin receptor substrate 1, it increases the affinity towards the tyrosine kinase domain of receptor. Clear? Means this presence of this SS2 domain leads to increase in the affinity between insulin receptor substrate and tyrosine kinase domains of receptors, right? Once it attaches to the tyrosine kinase domain of receptors, it gets activated by the phosphorylation event, right? It means first it gets phosphorylated, and this phosphorylations is influenced by the tyrosine kinase domain, right? That is present in insulin receptor tyrosine kinase, right? So now this phosphorylation leads to the activations of insulin receptor substrate 1. Once it gets activated, it undergoes the downstream or activations of downstream signaling cascade. So what is the downstream signaling cascade? This is the one signaling cascade and this is another signaling cascade, right? So in this signaling cascade, it first phosphorylates the RAS proteins, and after the phosphorylations of RAS proteins, how it is phosphorylated? Like previous ones, the RAS has also the higher affinity towards the IRS. So due to the higher affinity of RAS, it binds to this IRS. Ultimately, it activates the RAS, and RAS activation leads to the activations of inactivated form of MAPK, that is mitogen activated protein kinase or MAPK, and phosphorylation takes place. And these phosphorylations of MAPK means the activations of MAPK, and this activated form of MAPK directly targets the cell nucleus, and ultimately due to the binding of MAPK to the nuclear gene, and it changes the transcription levels and ultimately changes the level of transcription factors called protein factors. This is one pathway. Now the same things that I can explain using another diagrammatic representations. Here you can see this is what is the exact pathway. See, this is the MAPK pathway. The activations of IRS by the phosphorylation sequence, and this activated form of IRS enters into the downstream signaling cascade. In this downstream signaling cascade, there is multiple numbers of proteins, right? Initially, this activated form of IRS activates the RAS protein, and RAS activation ultimately allows the activation of other proteins that is involved in the MAPK pathway, right? So, what are the other proteins like GRB2, SOS? These are some other proteins where phosphorylation takes place in presence of the activated form of RAS, right? So, once all those signaling molecules gets activated, it activates the MAPK. Actually, MAPK is the kinase proteins, and activated form of this MAP kinase ultimately triggers the nuclear gene translations, and that changes the protein levels. And these protein levels, here you can see what is the most important proteins here. This is the GLUT4, right? Means once MAPK targets the genes, there is alternation of gene expressions. So, alteration of gene expression means it will produce some translated product. This translated products are nothing. Here it is the transporter. So, what is the transporter here? This transporter is GLUT4 transporters. This is very, very important. Why it is very, very important? Because GLUT4 is very specific for the insulin hormone, right? Now, this GLUT4 will allow the this GLUT4 is very, very specific for glucose, right? It allows the glucose molecules into the cells. Now, another pathway that is mentioned here, this is the IP3 pathway, actually second messenger systems. So, this phosphorylation of IRS or activation of IRS not only activates the MAPK pathway, this also activates this pathway. So, in this pathway you can see what are the most important signaling molecules involved. Protein kinase B or inactivated form of protein kinase B gets activated by the phosphorylation events, and once protein kinase B gets activated, it regulates some metabolic enzymes. So, like the enzymes that involve with the glycolytic events, the enzymes that involve with the gluconeogenesis events. So, there are several types of enzymes are synthesized in presence of the activated form of protein kinase B, right? And this activation is fully depends on the phosphorylations and activations of IRS, which is depends on the tyrosine kinase domains present in insulin tyrosine kinase receptors. See here, the things are briefed here also. You can see the transcriptions leads to the formations of proteins, and these proteins have several effects like cellular proliferations and differentiations. You also can see in some cases there is a degradation events of these receptors. See what is the role of this GLUT4? It allows the glucose molecules, and ultimately it undergoes cellular respiration events. And here you can see the second pathway that I have discussed here, the second pathway, this one. What is this? See, the phosphorylations of IRS ultimately activates the PI3K molecules, that is phosphatidyl inositol kinase, right? Phosphatidyl inositol kinase. And this activation is fully depends on the phosphorylation event, and this phosphorylation is carried out by the activated form of IRS1. Once PI3K gets activated, it activates or it phosphorylates the phosphatidyl inositol, that is 4,5 phosphatidyl inositol 4,5P2, that is present in the membrane, right? And phosphorylations of PIP2 ultimately phosphorylates next membrane molecules, that is PIP3, and these phosphorylations ultimately acts on the PDK, right? And activations of this PDK or recruit some other signaling molecules, that is AKT, and ultimately all these signaling molecules all these signaling molecules actually involves the phosphorylation events, and phosphorylation leads to the activations of those molecules, and this activated form of this signaling molecule alters the expression patterns of the genes, and alterations of this expression pattern of genes ultimately produce different patterns of protein, and that proteins will act into different levels in a particular cell types. Clear? So, this is what is the MAPK pathway, this is what is PI3K pathway. Now, in both the case, what is the most important things? The most important thing is the synthesis of formations of GLUT4, which is very, very important for insulin hormone actions, or this is important for the cells. Another important thing that is here is the rule of proper binding of insulin and the autocatalytic property of insulin tyrosine kinase. Now, here you can see the sequential events, how actually this pathway gets activated, and what are the terminal part of this pathway, that is it improves the glucose uptake by producing the GLUT4, and it also involves the glycogen synthesis because it promotes the synthesis of certain types of enzyme that is required for the synthesis of glycogens. At the same time, it also produces certain types of enzymes that promotes the biosynthesis of proteins. So, this is what is the importance of this signaling cascade. Now, the next part that is about the physiological functions. As I have already mentioned, what is the most important target site and what are the most important functions that is carried out by insulin hormones. Insulin plays an important role to keep plasma glucose value within a relatively narrow range without throughout the day, that is by maintaining the glucose homeostasis. Insulin's main sections are in the livers. Insulin promotes the glycolysis event and storage of glucose as glycogens called stored glycogens, as well as it can also involve to the conversions of glucose to triglycerides, because all of these act as the important source of energy. In muscle cells, insulin promotes the uptake of glucose and its storage as glycogens. Again, important sources of storage energy. In adipose tissue, insulin promotes uptake of glucose and its conversions to triglycerides for storage. So, ultimately we could say this insulin hormones very important because it in all the cases what we have said, it promotes the storage of glycogens, means it promotes the formations of energy, which is essential for cellular functions. Now, up to this what I have discussed is I have discussed what is the most important glands that secretes insulin hormones and other hormones like glucagon and somatostatins and some pancreatic peptides along with ghrelins. And after that, I have discussed about the maturation events of insulin hormones, and after that I have discussed about the regulatory mechanism of insulin secretions, and finally I have discussed about the signaling cascade where two different pathways are signaling pathways are involved that helps to regulate the metabolic activities and also involves with the formations of different type of transporters, which is essential for cells. And finally, I have discussed about what are the most important functional features of insulin hormones, what is their target site, up to which this is all about the insulin hormones and their role in physiological cell. Thank you.